Casey Burke. So in this case connection, I will be looking at case 2 and 3. Case 2 has to do with regulation of glucose, so I will be looking at the brain, the skeletal muscle, the liver, and the adipose tissue to understand the regulation of glucose. So when you consume carbohydrates, the glucose absorbed from your intestines quickly raises your blood sugar levels. In response, pancreatic beta cells detect this increase through a process that depends on the GLUT2 transporter and release more insulin. Insulin then binds to its receptors, which boosts glucose uptake in skeletal muscle, fat tissue, and the heart. This is primarily due to the rapid movement of GLUT4 transporter vesicles to the cell surface. Additionally, insulin suppresses the production of new glucose in the liver. So if we look at the skeletal muscle, glucose can be transported into skeletal muscle through both insulin-dependent and insulin-independent mechanisms. These include facilitated glucose transporters such as GLUT1, 3, 4, 5, 8, 10, 11, and 12. Once inside the muscle cells, glucose is phosphorylated to form glucose 6-phosphate, effectively trapping it within the cell. This intracellular glucose can then be either metabolized for energy or stored as glycogen. Next, I will be looking at the liver. The liver gets carbohydrates from food we eat through the portal veins, which carries them directly from the intestines inside the liver cells. An enzyme called glucokinase converts glucose into glucose 6-phosphate. This conversion ensures that glucose can enter the cell and be used for various metabolic processes. Glucose 6-phosphate can then follow several different pathways within the cell. Next, in the adipose tissue, it consists of fat cells known as adipocytes. In these cells, glucose transporter 4, also known as GLUT4, is present both in the cell membrane and within the cytosol. When GLUT4 moves from the cytosolic vesicles to the cell membrane, it increases glucose uptake. Conversely, endocytosis returns GLUT4 to the cytosol. Lastly, we'll be looking at the brain. So glucose enters the brain cell from the bloodstream by crossing the blood-brain barrier via facilitated diffusion, which is mediated by glucose transporters. In brain cells, glucose is crucial for energy metabolism, providing energy by generating ATP. So we just learned about normal glucose regulation. So how does this differ from tumor cells or cancer cells? So one of the key characteristics of cancer cells is their increased reliance on glucose for energy through a process called aerobic glycolysis. This process helps them produce the building blocks needed for rapid growth and supports nutrient signaling. It has been observed that cancer cells prefer glycolysis to generate ATP even when oxygen is available. In contrast, normal cells primarily use oxidative phosphorylation to produce energy and only switch to glycolysis when oxygen is low. Aerobic glycolysis is also known as Warburg effect, leads to the production of lactate even in the presence of oxygen. While this method is less efficient than mitochondrial respiration, cancer cells use it for other benefits. Researchers have found that this pathway supports important signaling processes that promote cancer cell growth and helps incorporate essential nutrients into new cellular material. Activation of certain oncogenes like RAS and SRC and transcription factors such as hypoxia-inducible factor 1 alpha can increase the activity of glycolytic enzymes and glucose transporters. So what might have occurred in cancer cells to increase glucose uptake as described just above? So cancer cells, as I said, require more glucose than normal cells. This increased glucose uptake is largely due to the overexpression of glucose transporters, particularly GLUT1 and GLUT3, which are responsive to low oxygen levels, as well as other glucose transport proteins, but GLUT proteins and sodium-dependent glucose transporters such as SGLT1 and SGLT2 are overexpressed in cancer cells. Additionally, cancer cells alter the expression of genes that code for HIF1, which is hypoxia-inducible factor 1, HK2, which is hexokinase, and PG, which is phosphoglucose isomerase, and glyceraldehyde 3-phosphate dehydrogenase, and various glucose transporters, which all play roles in the adaptation to hypoxic conditions within tumors. So to begin, case 3, this deals with a 16-year-old female who was recently diagnosed with a deficiency of muscle glycogen phosphorylase. Through my studies, I found that this is also known as McArdle's disease. So she has a few questions. She wants to know 4 questions, one of them being that she really likes to take walks with her friends, and she really wants to know if she's still able to do that. So if I were her care provider, I would say, yes, you are still able to do these long walks with your friends. Your condition, known as McArdle's disease or glycogen storage disease type 5, primarily affects high-intensity anaerobic exercises that rely heavily on muscle glycogen for energy. This disease results from a deficiency or absence of the enzyme muscle glycogen phosphorylase, which is crucial for breaking down glycogen into glucose 1-phosphate. This glucose 1-phosphate is then converted into glucose, the primary energy source for muscle activity. While McArdle's disease can cause symptoms like painful muscle cramps and weakness during physical exertion, your body uses aerobic metabolism for long walks. Aerobic metabolism relies more on fatty acids and blood glucose for energy rather than muscle glycogen. As a result, you should be able to engage in low to moderate intensity activities such as walking without any significant problems. However, you may experience fatigue more quickly than others, so it's important that you pace yourself and take breaks as needed. After initial fatigue, you may experience a second wind, where your symptoms improve with continued moderate exercises. Studies suggest that moderate intensity graded aerobic exercises can be beneficial for people with McArdle's disease, as it can reduce exercise intolerance and bring about the second wind phenomenon sooner during activity. Additionally, consuming simple sugars right before exercise, such as a sports drink containing sugar, might also help manage your symptoms and improve your exercise tolerance. My next question is, do I have to worry about having reduced muscle glycogen stores? So my answer would be yes. Reduced muscle glycogen stores can be a concern. When glycogen levels are low, your muscles cannot produce APT rapidly enough to sustain exercise intensity, leading to a fatigue. So some specific issues to be aware of is you might experience fatigue, muscle pain, and cramps within the first few minutes of exercise due to limited energy availability. Prolonged or intense exercise can lead to muscle damage because your muscles can't access enough glycogen to meet energy demands. And muscle breakdown during intense exercise can release myoglobin into the bloodstream, causing red or brown urine, so be aware of that. And also, ongoing energy deficits and muscle damage can result in lasting weakness, particularly in the thigh muscles or other areas. Her next question is, as a result of this deficiency, will I need to get up in the middle of the night to maintain my glucose levels? So my answer would be is unlikely that you will need to get up in the middle of the night to maintain your blood glucose levels. This is because your liver stores glycogen, which can be broken down into glucose to regulate blood sugar levels during periods of fasting, such as overnight. Maintaining a diet with high in carbohydrates can improve your exercise tolerance and overall energy levels. Research shows that carbohydrate-rich diet can help individuals with McArdle's disease perform everyday activities more easily and reduce the risk of exercise-induced muscle injury. For example, consuming prescribed amounts of sucrose before exercise can provide an immediate source of energy, helping it to manage symptoms during physical activity. So as I said before, all of that is very good and will hopefully limit you having to eat at night. And then also, Just having a good diet, certain supplements such as creatine, branched chain amino acids, and vitamin B6 may help with rapid fatigue, as well as and like maintaining your blood glucose level, as well as medications like ACE inhibitors or low dose intermediate acting insulin to help your condition as well. So you will not have to maybe do that at night, depends on you. Her next question is, do I need to worry about producing excessive lactate during intense aerobic exercises? So no, you do not need to worry about producing excessive lactate during intensive aerobic exercises. And McArdle's disease patients have a marked decrease in plasma lactate concentration at the onset of exercise, and this low concentration remains constant during exercises. This condition results from a deficiency in muscle phosphorylase, which disrupts glycogen metabolism at the beginning of glycogenolytic cycle, preventing the normal increase of lactate levels during or after exercise. So, lactate is an important energy source for contracting skeletal muscle. Individuals with McArdle's disease do not experience a significant increase in lactate levels during exercise.